Okay, so I think we're good to start. So we will continue with cancer genomics. Uh, Thank you, Shilpa, for being here and for your presentation. So Shilpa is now an associate professor at University of Manchester. She was a research fellow at Harvard Medical School, and she finished her PhD in computer science at Max Planck Institute. She worked in many different fields of bioinformatics and computational biology, and she likes to apply the power of computer to understand the biology of complex disease and sustainable utility analysis. And today she will tell us about, as you can read, towards routine chromosome scale haplotype resolved reconstruction in cancer genomics. So please feel free to ask any question either in the YouTube chat or in the Matrix channel as the talk goes on, and we will answer them to the speaker at the end. Thank you again, and please, the stage is yours.
Thank you, Sumani, for the kind invitation, and nice to meet you, everyone online. I hope you're enjoying biohacking. So I'll give you some insights and journey towards chromosome scale haplotype resolved reconstruction in cancer genomics. So here is so before I delve into the scientific topic, I would like to introduce my team. So we are a set of um, people with diverse cultural backgrounds as well as interdisciplinary skills. And we are excited to contribute to the development of new computational approaches that harnesses cutting edge technologies like long reads, high C, single cell to address societal challenges in sustainability and health. So more specifically, we are interested in pangenomic methods, so that aligns very well with the aim of today's biohacking. And we are excited about graph-based methods for integration of multiple biological levels, structure variation discovery, and single-cell multiomics. And now here is the outline of my talk. So I'll walk you through some of the methods that we have developed for reference-based phasing, and then how we generalize to more chromosome scale haplotype resolved assembly of genomes, starting with some simple like normal human cells where it's deployed, but then slowly I'll give more insights into more complex genomes like cancer genomes and their clinical relevance. But for today's talk, I'll skip sustainability angle. Um, but so, as you know, humans are diploid, so there exists two copies of chromosomes, and we know the genotypes AT and CT of these variants, let's say. But the thing we want to know are the haplotypes, like A is connected to T and T is connected to C. And the process of obtaining this information is known as haplotyping. It has many applications, like it helps to study human migration, evolutionary structures, and population genetics. It also helps to understand causes of diseases. So there have been various approaches to phasing, like molecular haplotyping, genetic haplotyping, and statistical haplotyping. So without going in much detail, I'll give a brief um, introduction to some of the statistical techniques that were used a decade ago for doing started a decade ago for doing the population based phasing. Essentially, you are given a, a population panel with a set of genomes, and then the idea is you want to infer the haplotypes of the target based on this common ancestry information that is hid that is embedded in this panel. So on the left in the x-axis, you see the size of different reference panels, 1,000 genomes or HPRC, and on the y-axis is the phasing error rate. So if you use about thousands of genomes, you get about like 1.5% phasing error rate. But then if you increase the sample size to about 100,000, then the phasing error rate dramatically reduces. So this suggests you always require a large sample size, and but then another limitation is you can't phase de novo or rare variants, and you can't really study the chromosomal scale phasing and HLA and KIR regions. So to overcome this limitation, here we proposed a read-based phasing where you take a reference genome and then you have these three variants, and you take the reads from the target genome and then you align these reads to the reference, and the alleles that are colored in red, these characters, they represent the alleles that cover these SNPs, and the goal is now, because of the diploid genome, you want to partition the reads into two sets um, such that there is no conflict in any set. Imagine when it's a perfect case, there are no sequencing errors, then it's trivial to solve because these reads are originally coming from two sets of chromosomes, two sets of copies for each chromosome, and then you can easily make these partitions because there are no errors. But in the real case, in the actual experiments, you see a lot of errors, especially like traditional long read sequencing. It produces about 5 or 10% error rates or even more. So now to solve this problem computationally, we formulated as a minimum error correction problem, so which is to flip the minimum set of entries such that conflict-free bipartition exists, and where conflict-free bipartition property holds if the alleles between any two rows in partitions match. For example, here if you flip this G and T to the other allele, you will start to see the make the partitions, and therefore you can assemble them to two haplotypes. So now we looked into combining different types of, as the technologies were advancing, like long reads or single cell, we looked into how can we leverage the technology advancements to produce more and more complete phasing or more better genomes. So we started looking at strand-seq and PacBio data. So strand-seq is a single cell technology, so you will see that it's a very sparse data, and then but the good thing is it's on the global scale, so it provides phasing on the global scale, indicating, for example, in this row we know that this read comes from one of the haplotypes, but then there are series of gaps here because of the single cell or sparseness uh, of this data type. And then we also looked at long read sequencing, for example, from PacBio, Nanopore, or 10x sequencing, which is a bit more dense. It gives a continuous series of informations of zeros and ones, but it's a bit more local, so it cannot traverse the whole genome. So we started looking at these instances, and we implemented the dynamic programming based algorithm to solve the MAC problem, which provides optimal solution and efficient solution. If you are interested to know more about the dynamic programming, we can chat offline or after this, or even now, since this is a discussion. If you have immediate questions, I would be happy to take. So moving on, so we performed experiments by combining different technologies like PacBio and 10x and many other combinations. So in this plot, you will see that each color represents a phasing segment, and this panel represents a chromosome. So you will see that more the colors are, more fragmented is the phasing. Uh, so if you ideally to get a chromosome scale phasing, we would like to see one chromosome, one color through the whole panel. And this white color represents the centromeres. So by combining different coverages of different data types. We performed several experiments, but we observed that even by taking all like high coverage data, we still can't get the chromosome phasing. But when we combine strand-seq with the 10x data, we for the first time can here produce a chromosome level phasing, suggesting that we can achieve now reference-based chromosome scale phasing with also very high accuracy. So we benchmarked against genome in a bottle, and also we used pedigree information. So here we see that the switch error rates and like the Hamming error rates are like 1%. So moving on, as we looked more closely into this analysis, so we observed that there is a reference bias here. So imagine you have a very diverse region in a genome, or you have an entirely new genome from a different um, genetic ancestry. So here, reference-based analysis will have some limitations. So to overcome that, we looked into a more de novo style um, genome assembly. So essentially, for to do that, we looked into hi-fi and Hi-C reads. So hi-fi uh, reads are highly accurate reads, and Hi-C are more on a long range and chromosome capture technique. I'll give a bit more introduction to these technologies now. So, in the hi-fi technology, you essentially start with the high quality molecular DNA, and then you ligate the adapters, which lead the primers to be annealed and bind the DNA polymerase. So, after this step, the DNA is now circularized, and then essentially you perform repeated passes of these sequencing and then call the consensus with highly accurate long reads that produces highly accurate long reads. And then looking into the Hi-C technology, which is more of a captures the spatial nature of the 3D structure. So instead of on a linear scale, it can be megabases apart, but on a 3D spatial space, this is quite close by. So this is exactly the information is captured using this Hi-C technology. So here on the right side on x-axis, you see the genomic loci. On the y-axis, you see the contact frequency. So darker the color is, higher is the contact map frequencies. But you will see on the top, you see less dark, less red color, so which indicates that the signal is not that good from the Hi-C data. So looking into genome assembly, how can we really combine these technologies to get high quality genomes? So initially, when this whole field started 3 decades ago, um, the the graphs were made using short reads, so therefore they were very tangled, and the sequences were discontinuous. But as we moved from short to long reads, the um, the sequences became more continuous and the graphs were less tangled. But for example, computational this problems has been formulated as two type of graphs, either divergent graphs or overlap graphs. So in the divergent, you break a read into k-mers, and then you find the continuous between these two, between the k-mers, and then essentially you find the haplotype paths through this graph. And in the overlap graph, in each read, each node is a read, and the connections are the overlaps between them. So, but in this era, still because the long reads were still um, erroneous, so therefore the haplotypes were still collapsed into a consensus and ignoring the diploid nature of the genomes. So as well as the phasing was quite fragmented. So to overcome this problem, we actually introduced and read overlap graph problem, which is to look into the phasing information right within the overlaps. So essentially, you find the overlaps between the reads, and then you look into the SNP information, which is quite powerful to tell you which reads comes from which haplotypes. And this information is encoded in the form of graph. So each node is a read, and the connections are based on the haplotype specific overlaps. So, and then you start to see these lovely structures in the graph known as bubbles. So each node bubble has a common start and end node, and then there are haplotype paths within the bubble. And these bubbles can also represent the structure variations and so on. So we proposed a workflow, which is how can we efficiently combine hi-fi and Hi-C data. So we first took the unfazed contigs, and then um, we bring the Hi-C data on these contigs, and then we get the unfazed scaffolds, and then we map back hi-fi reads to these unfazed scaffolds to get these snip calls. And then we use these phase snip calls to really make the partitions and finally get the two haplotypes for each chromosome. And we, for the first time at that uh, several years ago, could do this within a day, um, and which used to take um, a month's time to assemble a genome. So, and then we benchmark this method on HC002 and many other public genomes available. And, and we observe here we can produce about 3 gigabyte bases of each haplotype, which is about same as the trio Kanu. And then we can produce better continuous assemblies, which with 25 um, megabases, and we can phase larger fraction of the genome about 8 times better than other methods. And and we perform the same experiments for other genomes as well, like NA12878 and PGP1, and we observed the similar trend. And then we also looked at few more clinically relevant regions like HLA, so which is a highly diverse region. And as you see, when we aligned our assemblies to the reference genome, each panel is represents the haplotype of from a for a given sample, and then the colors represents the amount of divergence of this specific region to the reference genome. So higher, darker the color is, higher is the divergence to the reference. So as you see, there is even about 10% divergence in some of the genes like DQB1, DOA1, and so on. So which we can reliably unravel only when we have the phased assemblies available. Next, even in the Key region, we observed that we could now reliably phase Key and produce whole gene, produce phased assemblies, and then there are there's a high variation of about 10% within this region as well. So moving on, when I started my own lab, I got excited about cancer genomics. So as we know, mainly to study tumor heterogeneity and the treatment resistance, and how these long range, how the variation is linked to promoter enhancer interactions. And I got excited to study the distribution of variants, that how affects regulation, and that two homologous gene, for example, if there is a variant here and in one of the haplotype, it produces a different phenotype in cancer than the other cases when there is no variant available. So there have been traditionally some tools available to study like the phasing of variants within cancer genomes. So like Lira, Monovar, or infer CNV. But these tools had limitation. Either it has a high false positive rate, or they don't consider the complete high base resolution phasing, and they can't work for complex genomes. So here I proposed a study design to look. We started looking at colo. There's a typo, sorry, 8 to 9, and we also collected the bulk data and we sequenced it using HiFi and HiC, and then we validated our findings using the benchmark data sets available. So there is a good match tumor normal available for colo 8 to 9 cell line, and then we sequenced this using HiFi and HiC technology to about 35x coverage for both tumor and normal. And then I proposed a new workflow. How can we integrate these new data types in the context of cancer genomes? So first we took the HiFi united graph, which was produced by HiFiasm, and then we called the we now map the HiC reads directly onto the HiFi graph by taking the the distinct k-mers that can occur between alleles in the bubbles into consideration. So most of the mappers, they were not able to take the heterozygous k-mers into consideration. So this is for the first time we were able to perform phasing aware Hi-C read mapping to the graph. And then essentially, first we produce the haplotype paths based on the maximum support of the reads. So now you get different components in the graph. So we map the reads. So first we solve the phased assembly problem for each component, producing the phased contigs. And then we connect these phased contigs based on the neighborhood property. Essentially, and we project it into a new phased scaffolding graph. So what's a neighborhood property? For example, for a given edge, if the neighboring edges have a comparable high C support, then we treat that edges as a true signal from the height C data. Whereas in the uppercase, the neighboring edges have only a support of 2, so these connections might be erroneous. And this was mathematically encoded within the method to then that was useful to produce now the phased Conte scaffolds for the genome on the whole genome scale level. So then we really were interested whether the phased scaffolds that we produced there on the chromosome scale level. So here, then we map them to the reference genome for both H002 and Colo 829. And on the left, you see each panel represents the chromosomes, and then there's one color through the whole chromosome for all chromosomes, um, so which represents that we can now produce chromosome scale phased assemblies for normal as well as cancer genomes like colo 829, which is a melanoma cancer. But there are still, in this case, there are still regions like acros that we haven't resolved in this case. So then using these assemblies, we actually got interested to call comprehensive variant landscape to study the comprehensive variant landscape. We looked into germline and somatic variations, and specifically we looked into the repeat families. For examples, different alus, lines, signs, L1 and L2. And we also looked at the concordance of our variants to the different benchmarking methods and data sets available. So the blue region represents the concordance of our uh, calls with the single cell data, which is quite high, which is more than uh, in line 1 more than 90%, whereas um, um, and then we also looked at the concordance of a method with other available short read based calls, like from one from the New York Genome Center, another one available from the Netherlands Center. So in some of the cases, um, we observed that there were some calls that were discordant, and then we again looked at their concordance with the single cell data, and there are actually some rare clones in the melanoma that we have missed, that were actually called by the single cell data. So a combination of long reads and single cell is the best to really study the structure variations at a higher resolutions, but which is um, but there are some experimental challenge. But at this point, so we can resolve 90% of the structure variation calls at a higher resolution with long reads. So we called about 26,000 somatic and germline structure variation calls, out of which there are about 13,000 insertions and about 9,000 deletions and 29 duplications. And we were also able to call P10 deletion, as you see, which is also on the both chromosomes that we could call, which is already known to be have an association with melanoma. So moving on to like beyond going from one genome, how can we really study cancer genomics or biodiversity? So to do that, we also started looking at chromosome scale haplotype resolved pangenomics. Essentially, one of the approaches is you take a reference genome and then you align different genomes to the reference and embed that into a graph. So each here these homozy the nodes represents the sequences in the graph, and but that is common across different genomes or across different haplotypes. And then there are these special structures in the graph like bubbles that are actually helpful to encode small variants or tandem repeats or even complex structure variations. As you see, if one of the genomes, especially in the cancer or other complex scenarios, if one of the genomes have a huge variation, that can be encoded in the form of this graph. So it has associations with helps to study the associations with gene expression, functional data, and also helpful to encode different informations like population frequencies and signatures. And beyond human health, this can be also helpful in sustainability angle as well. So traditional pangenomes were more like either a set of genes, mainly in the microbial field, and then and also in the human field, it's more like you collapse the genomes, and then the genomes are quite fragmented, and these were essentially it's a collection of contigs from different genomes. But the graph-based methods can actually reliably encode these reads. So first is you take a reference, you take the reads from the target sample, and then you start adding the variation one by one to the reference. And the other is the chromosome scale, really working on the chromosome scale sequences, phase sequences. So there are different methods like Kamer keys or minigraph or wavefront alignment algorithm that could be helpful to produce a graph pan-genomes. But these techniques have some limitations, like they are not able to either encode the whole evolutionary structure that can occur in different clones, in cancer genomes or in biodiversity. So we actually proposed a hierarchical approach instead of embedding the whole variation at one go in a big graph, but take a more hierarchical approach. So we divide the evolutionary clones in the form of, you can imagine them as like species, genus, class, and then each level has the set of sequences, and then you really look at the most diverse sequences at each level and encode them in the form of a graph. So here each subgraph represents a, a level in this hierarchy, and then each node in a graph is the is the sequence that is um, in the set of sequences, and then the most diverse sequences will be represented in the form of a bubble in the graph. So now you can encode these information across um, across a hierarchy rather than every. So there are advantages to this. So it can be easily accessible, it could be highly scalable, but there are disadvantages that there are, if there is a 90% diversity, then we cannot represent this whole information easily. So with that, thank you very much for your attention, and I would like to thank my group and the funding agencies, and thanks organizers for inviting me.
Thank you very much. Thank you, Shilpa, for this lovely talk. Let me see if we have any questions in the chat. Looks like we're done for now. While we wait, I do have a very broad question. I saw most of your work now is being done on these long reads. That for a long time we kind of considered not very reliable, right? And we had to also rely on short reads to make sure that we have a good quality. So I saw that with these new technologies such as the hi-fi, now these are quite good. So do you think, given the opportunity, it's time to move from short read to long read, or are there still some requirements that still we need to address when we use long reads?
That's a great question. Thank you. So it depends on the application you have. So, for example, in cancer, um, and also the cost considerations, and there are many factors in this. But if you have like long reads are getting cheaper, that's one thing. Another is like the high molecular weight DNA. So if you have a fresh frozen sample or a liquid biopsies available, then I think long reads are quite feasible to perform. Like if you have high quality like um, molecular DNA available, I think then I would. So in different, so I would pitch moving towards the long reads if like the requirements are fulfilled to achieve a high quality data, especially, and also the price is now getting like $500. Hopefully in a few years it will drop down to $100 or so, even beyond. So because there is a lot of value, short reads, we were seeing like breakpoints of SVs, and still the analysis requires a long time. But now if you are interested to have a comprehensive landscape including in repeat regions on the human genome, I would like, I think the community, and I would like to see more and more long reads and high C in the field.
Okay, thank you. And since you mentioned right now SV, so structural variation, do you had opportunity to see them applied to this type of pangenomics? Do you think we we do have something? Because I know there are a lot of people working on that, but it looks like integrating them into pangenomes is quite difficult. Either they do like pangenomes for structural variations, or try to include them into like a normal pangenome also had structural variation is a big challenge. Do you have any experience with that?
Yeah, I think I understand and kind of agree with you. Like this is an evolving field. Like we have seen a high resolution pan genome just a few years ago. So it requires a whole new machinery and computational tool development, and to really mature to a level when it becomes like a routine thing. So it we have taken, the community has taken some initial steps to establish such a machinery that we can do structure variation calling on the pan-genome, but I would say it's still developing more. Furthermore, developments are required. So there is a great effort in US, which are also a part of human pangenome, so which will run again other like few years and then 5 years, and then hopefully like this will be a great ecosystem. And also there are many projects in Europe that so this is an evolve. Hopefully, in another few years, we will solve this problem more efficiently and effectively.
Thank you very much. So it looks like our chats are a bit shy. With that, I would like to thank you again. So we will have a smaller break.